Good afternoon, everybody. Before I begin, I'd like to thank my collaborators on this project for their guidance and support, and also my awesome lab group for their hard work. Our group, the Movement Ecology and Reproductive Resilience Lab at the Fish and Wildlife Research Institute, has conducted a 3-year study on gag grouper in the Madison-Swanson Marine Protected Area, and are now finishing up a second 3-year study in the Steamboat Lumps area. And before I click on, I'll give you a moment to appreciate these beautiful gag. So to begin, I'll briefly review the metapopulation concept. A metapopulation is defined as a regional group of connected populations of a species. In the schematic, circles represent populations, green arrows indicate exchange, and gray arrows local self-recruitment. As local populations fluctuate in abundance, they may become vulnerable during periods when their numbers are low. The regional persistence of these vulnerable groups is dependent on the existence of an interconnected metapopulation. Elimination of metapopulation structure through interference with exchange can result in population declines and a net loss of resilience. The following excerpt illustrates this. It is the degree of demographic connectivity that characterizes metapopulations. With the dynamics of local populations strongly dependent upon local demographic processes, but also influenced by a non-trivial element of external replenishment. Therefore, estimating rates of interpopulation exchange must be a research priority. The existence of movements between subpopulations confers metapopulation resilience, and variation in these movement patterns augments this further. Contingents are groups within species that exhibit different movement patterns, and diversity of these patterns results in increased resilience against spatially explicit stressors in a manner analogous to a genetic portfolio effect. In other words, greater diversity in behavior provides multiple avenues for metapopulation persistence. Even if one exchange pathway is disturbed, others persist between subpopulations. West Florida shelf gag have a particularly complex spatial life history with sex-specific movement patterns. They are protogynous hermaphrodites, meaning they're born female before ultimately turning male. And larval gag settle in West Florida estuaries. Subsequently, female gag reside in nearshore waters, forming pre-spawning aggregations in the wintertime before migrating to offshore spawning sites. The sex transition occurs later in life, with 50% transitioned by age 12. Male gag reside at these offshore sites year-round, and these spatially isolated groups are linked through migration and dispersal. In recent years, fishermen have failed to meet their quotas. Spatial management measures, specifically marine protected areas, designed to protect spawning aggregations, were implemented to improve the gag sex ratio from roughly 2 to 3% in the 1990s, but without success. And this suggests that gaps in our understanding of gag population connectivity have led to ineffective spatial management. If we don't know the traits driving reproductive success in gag, specifically their movement patterns, then we probably are not managing to protect them appropriately. Evidence exists for genetic differentiation indicative of potential subpopulations between Campeche bank gag and West Florida shelf gag. And in future work, I plan to use higher resolution genomic techniques to assess such differentiation on a smaller scale, specific to the West Florida shelf. Until recently, metapopulation structure was difficult to assess using genetic markers like those mentioned in the previous slide, because interdependence between subpopulations occurs in regions of high migration, and we did not yet have techniques with the requisite high spatiotemporal resolution. However, the processes and behaviors that compose this metapopulation structure can be examined. Here we will look at dispersal and connectivity for multiple spawning sites, for example, Madison-Swanson and Steamboat Lumps, which are spatially isolated reproductive hotspots, in order to better understand gag metapopulation structure and resilience. Gag complex movement patterns relevant to the metapopulation concept break down into migration and dispersal. Their adult migration is comparable to bird migration, And like in seed dispersal, gag embryos are released en masse to be ferried to their ultimate settlement destinations by external forces. In combination, though, this results in a complex system that is further complicated by larval behavior post-hatch. Earlier studies of population structure cite larval dispersal as a connector of gag putative subpopulations. And the importance of spawner recruit behavior in space and time, and specifically larval dispersal, is best summed up in the quote shown here. Although reproductive success is tightly coupled with adult abundance and fecundity in many terrestrial animals, in exploited marine fish, where and when fish spawn and consequent dispersal dynamics may have a greater impact. We set out to investigate the following areas, focusing on larval dispersal in particular: to elucidate patterns of dispersal-driven connectivity and their variation on an ecological timescale, and to understand how the complex interactions of gag embryonic and larval traits with their physical environment drive variation in spatio-temporal trends in settlement and survival. To achieve this, we are using the Connectivity Modeling System, or CMS, the Lagrangian framework, which exposes simulated particles to a three-dimensional ocean velocity field pulled from the Hybrid Coordinate Ocean Model, or HYCOM, and maps their trajectories over space and time. To improve the model's realism, we incorporate known spawning sites, reproductive behavior, including timing, frequency, spatial spread, and relative magnitude, as well as embryonic and larval traits. However, it's worth noting that much of the data needed for this table is not yet available for gag grouper, necessitating the use of data from other species. And part of my dissertation will encompass filling some of these data gaps. Here, circles indicate known spawning sites at which particles are released in our simulation. Large red spots indicate high activity sites, while smaller blue ones have lower spawning activity. So here are preliminary model results. The black points indicate release locations from which simulated particles are deployed, shown on the previous slide. Red points represent locations at which these particles reach suitable settlement habitat. Colorful trails indicate the trajectories of these particles throughout the simulation, and their colors represent the time since each particle was released. The percentage of particles released in a given year that reached suitable settlement habitat within the pelagic larval duration was tabulated and included as the survival metric in each pane. Red boxes around this value indicate particularly low survival years. In green box years, high abundances of age 0 gag were recorded in West Florida estuaries by the FWRI Gag Juvenile Fishery Independent Survey. And the take-home message here is that reproductive success is complex, as embryos and larvae need to arrive at appropriate settlement sites, even though annual oceanographic conditions differ. Our preliminary results expose questions for future investigation. For example, we don't yet know if adult fish alter their reproductive behavior to address changing oceanographic conditions, and or how larvae change their location in the water column, and thus their resulting dispersal patterns, depending on those conditions. In our ongoing work, we'll focus on the following items. First, iterative improvement of this model through the incorporation of new reproductive and oceanographic data, and ground truthing through comparison with regional trends of model recruitment and age 0 gag abundance metrics in West Florida estuaries. Following continued improvement, the model will serve as a predictive index of recruitment with utility for spatially explicit stock assessment. In my doctoral research, I plan to incorporate a natural biogeochemical tracer component to understand connectivity between estuarine gag nurseries and spawning sites, taking our understanding of the gag life cycle full circle. And also genetic analyses, as previously mentioned, to quantify the structural effect of gag population connectivity patterns over an evolutionary timescale. Together, the model and my other dissertation research will inform spatial management strategies for gag grouper. I will integrate a high-resolution understanding of West Florida shelf gag adult reproductive behavior with larval behavior and survivorship in the context of metapopulation resilience, ultimately helping us better understand which aspects of GAG spatiotemporal spawner recruit behavior affect reproductive success and thus warrant protection. Thank you for listening and have a great day.